So moving on to our next speaker, Professor Phil Beales needs no introduction to this group. It was in June, exactly 25 years ago, that Professor Beales and colleagues published the diagnostic criteria for Bardet-Biedl syndrome, supported by Drina Parker and BBS UK, or the LMBBS as we were then known, and those diagnostic criteria are still in use today. In 2010, Professor Beales established our NHS-funded multidisciplinary clinic service, including BBS UK as a contracted partner. And as Shella has just said, this service has transformed healthcare for our group, reducing the age of diagnosis and facilitating an improved knowledge and understanding of the syndrome. Professor Beales has been medical advisor to BBS UK since 1996, president since 2005, and is a founder member of the BBS UK Scientific Advisory Board. Is joining us today to provide an update on research and study of Bardet-Biedl syndrome.
All right, thank you. So it's great to see so many people online. I'm sorry we can't all be there in person, but in a way, we're reaching out to a lot of people. I see, just looking at the attendance list this morning, that going west, we have Mohammed from Portland, who is a trooper. He's up at 2:30 in the morning. Welcome. And Gina's next in New York. Must be getting almost on for breakfast now. And I see Ali in Iraq, who's probably already tucking into lunch. So everyone's welcome and really pleased to be here today. So what I have been asked to talk about is give an update really on the therapeutics that may be coming our way. And it's something that I am absolutely delighted to be able to talk about, simply because we have not, well, I never thought that we would see treatments in my lifetime. And I'll explain what I mean by those treatments, because there are treatments and there are treatments. So just beginning with some of my disclosures. So for example, I am not only the co-founder of Axovia Therapeutics, I'm also an employee of Axovia Therapeutics, which is the spin-out startup company that has come out of UCL back in 2020, and that is to develop the therapy that I'm going to talk about. And as Tonya has mentioned, I'm also medical advisor to BBS UK. So thank you, Shella, for describing so eloquently the background to Bardet-Biedl syndrome. And we don't need to obviously cover those again, but simply to say, and you've already realized this, that so many parts of the body, what we call so many organ systems, are affected. And for us, for a long time, certainly in the early days when I was researching this condition, we had no clue what could it be that was actually underpinning and causing all of these different problems in different parts of the body. And what we did know, though, that there was, at the time when we were first looking for the first gene, we thought that maybe there was just one gene. And again, as Shella has said, there are more than 22 genes. In fact, it's probably thought that about 25 genes now and counting. But the good news is that the majority of those genes that have been discovered are actually providing us with robust genetic testing for probably well over 90%, or at least 80% of the patients that come to see us in clinic. So again, I don't want to delve into the details of the clinical diagnosis because Shella has explained those. But what I do want to say is that when we did publish the first, back in 1999, the first diagnostic criteria, these were really laid down because there was no alternative. There was no genetic testing, no gene had been discovered. So this is really very much the 20th century approach to diagnosing Bardet-Biedl syndrome. And it still has a role because it is suggestive of this particular diagnosis, which can now, thankfully, be confirmed by genetic testing. And so therefore, if these set of criteria do turn up, then this should suggest to clinicians to go out and test for Bardet-Biedl or ciliopathy type genetic mutations as such. However, we are beginning to see, just in the last 3-4 years now a number of patients who do not fulfill these criteria, and therefore, but also do have a molecular diagnosis as such. They're not that many, but there's still quite a few. So this tells us a couple of things. First of all, that these diagnostic criteria perhaps ought to be modified, and I know that several groups are modifying them at the moment. And secondly, we have to consider where what is the role of molecular testing in this particular situation. And it also tells us that there are plenty of people around who do have two mutations in a bona fide Bardet-Biedl syndrome gene, but do not necessarily have all of the elements of the condition. And so I can tell you in our clinic, for example, we get a lot of referrals from the eye hospitals where patients have been seen for many years and diagnosed as having retinitis pigmentosa. And it turns out with subsequent genetic testing, as that's become available, that they actually have Bardet-Biedl syndrome, and then they come to our clinic. Again, as Shella has mentioned, we showed a number of years ago that Bardet-Biedl syndrome is caused by cilia dysfunction, these little finger-like appendages that come out of the surface of all the cells in our body. And really a lot of the symptoms that we see, symptoms and signs as we call them in medicine, relate to where those cilia are and how they have failed, if you like, in their primary function. So for example, I'm showing you in the top here, let me just get my pointer up. In the top here, these will be the type of cilia that you'll find in your lungs, in your breathing tube, the so-called trachea, and they move, they beat, and they essentially have to move fluid around and clear out the phlegm and various other particles that we breathe in every day. And of course, this can present in some patients with BBS as recurrent chest infections. Again, that can easily be related to the function. However, there are other cells in the body that don't move, other cilia on cells in the body that don't move, such as in what we call the olfactory apparatus, which is obviously in the top of your nose and allows us to smell. And those cilia, they don't necessarily beat, but they do receive the chemical that we then translate into a particular smell. And so we now know that many patients with BBS have a reduced or complete inability to smell very well. And then, of course, the biggest area where there is an impact on the failure of cilia is in the eye. In fact, in the back of the eye, the retina, the so-called photoreceptors, which contain a cilium. And of course, if this cilium is not working properly, then it leads, as we well know, to degeneration of those photoreceptors and visual loss. So I'm not going to go into it, but BBS has really led us to understand a lot of other ciliopathies out there. There have been many others that have subsequently been ascribed to cilia dysfunction, probably about 45 different conditions at the moment that we know about, and they have similar overlapping features, which I won't go into today, but they're very important collectively because they affect around 1 in 1,000 of the general population. Another important discovery that was made some time ago, back in 2007 now, by Max Naturi's group and Peter Jackson in the States, was essentially the discovery of the BBSome. Now, this is a complex of proteins that essentially sit at the bottom of that cilium, that finger-like appendage that pokes out of the top of cells. And its main function is to move proteins in and out of the cilium and also around the cell. And we now know that if there is a genetic mutation or two genetic mutations that any individual might be carrying, this can affect the function of this very tightly knit complex called the BBSome. And we can see a number of different genes that are represented, or gene proteins that are represented within this complex. There are 8, in fact, that are listed here. I won't go into them just at the moment. But the fundamental point here is that if this BBSome fails, because you do have mutations in any one of those genes, then it can lead to visual loss, as I've just said, because the photoreceptor, as I'm showing you in this diagram here, consists of 2 parts, an outer segment and an inner segment. And all the proteins are made in the inner segment, but have to be transported to the outer segment for vision to occur successfully. And this essentially breaks down with time. And we know that because the age at which children start to lose their sight starts quite early, with the loss of rods in the back of the eye, leading to night blindness. And then eventually daytime vision will go sometime in the 2nd to 3rd decade, due to the fact that these cones also start to die as well. So therefore, how might we consider starting to manage these kind of genetic conditions like Bardet-Biedl syndrome? So for us, for the longest time since we've started these clinics back in 2010, we've been really addressing the symptoms because there were no fundamental root cause treatments out there. So for example, if someone has high blood pressure, we'll put them onto an antihypertensive medication. If they develop diabetes, of course, we will treat that. Depression might also have obviously behavioral therapies, but also drug therapies as well. And for the longest time, the only way in which we could manage weight gain, and perhaps some would say not very successfully, is through diet. And we now know there are many other drugs that are coming to the market now that could also be beneficial. But all of these are essentially treating the symptoms. They don't actually get to the root cause of the problem. And of course, for vision loss, we all know that there is no treatment at all that's available. However, we are also interested in looking at the cause. Are there treatments that we could perhaps scratch beneath the surface, if you like? So of course, organ failure, kidney failure being one of those, we could get at that root cause, if you like, by doing a transplant. And that's a well-known therapy, renal replacement therapy that is done all of the time. And the next, as we've already described, is ciliary dysfunction. Could we restore ciliary function? Well, very unlikely to be able to do that directly, but we could do it indirectly by replacing the missing protein, in this case, BBS. And that's not so easy. There are other diseases out there, such as enzymes, where there are enzymes missing and you can replace those enzymes. But in our case, it's going to be much more difficult to do that. But we can replace a gene, if you like, or augment a gene that is dysfunctioning. And this is exactly the approach we've been taking for Bardet-Biedl syndrome. So very briefly, gene therapies fall into 2 different categories. Fundamentally, they all involve packaging either DNA or RNA, the genetic material that we want to replace, into a vector. And that vector could be a virus, or it could be a liposome particle, if you like. And of the 2 types there's what's one called ex vivo, which we're not really going to talk about today, and in vivo. Ex vivo is where you take the cells from somebody who has that particular condition and reintroduce the genetic material into those cells in the lab, and then reintroduce that those reverted healthy cells back into that person. So this has limited application, but it's very good for blood diseases, and it's been used very effectively to cure diseases such as immune deficiency, the boy in the bubble, for example. But the other approach, in vivo, which I'm going to focus on now, is really about genetic material being delivered directly to the target organ that has failed. And you can either do this by injecting directly into that area, or giving it intravenously. So those are the key approaches at the moment. And so we've decided that because, as Shella mentioned earlier on, the most commonly represented, at least in the West, gene that is mutated in BBS is BBS1. So we have developed a BBS1 gene therapy. I won't go into the component parts of that, but it's quite complex. But we have to move this gene with all its other component parts into and package it successfully inside a virus. Now, this is a deactivated virus, so it doesn't have any, it doesn't cause disease in humans at all, but we sort of hijack it, if you like, and put the DNA of interest, in this case, BBS1, into this virus and use that to carry the DNA into the cell of interest. And in essence, what we are focusing upon is delivering this to the back of the eye, to the retina. I'll show you how in just a moment. So once this enters the cell, in this case, it's going to be a photoreceptor, the virus will then dissolve and deliver its package, its payload, into the nucleus. And once it's in the nucleus, it will essentially start to produce what's called mRNA, which is the precursor or the blueprint for proteins. And then the proteins are made in the cytoplasm, or the body of the cell, if you like, where it stays and does its function and helps cilia to function, for example. So that's the background. So let me show you some of the data that we have been recently sharing with the world and has been shared at the American Society of Cell and Gene Therapy just last month by my friend and colleague, Victor Hernandez. And all the work I'm going to show you is in our friend, the mouse. And we're very fortunate to understand that the mouse actually has, well, recapitulates the genetic forms of Bardet-Biedl syndrome, but in particular, early onset weight gain and early onset loss of vision. So we can use these as our test organism, if you like. And so very briefly, we've divided the study, this is the description of the study groups, into 10 different groups. And we compare essentially the mouse that has BBS with their what we call wild type or normal mice that do not have BBS. And we want to test 3 different doses. We've tested low dose, medium dose, and high doses of the particular vector that we have made, of the BBS1 vector. And we deliver this over a 6-month period, and we measure, just like we do in patients, the ERG, which is the signal, the electrical signal from the back of the eye going into the brain. And we measure the thickness of the retina using a measure called OCT, optical coherence tomography, which a lot of you will have had if you've ever been to the opticians. And so how is this done? Well, simply we inject, I say not simply, it's not simple. It has to be done by a specialist surgeon, and is very highly skilled. But the simplicity is that you're delivering the gene therapy into the space around what's called the subretinal space around the photoreceptors. You want to get as much of this gene therapy to the photoreceptors themselves to be able to see an impact. So the first question we ask, and this is a complicated slide, but it isn't really, is does this approach using this virus, is it actually going to deliver what we need to the cells that matter, in this case, photoreceptors? And so in the first experiments here that we're showing on the left-hand side, or these colorful images here, they are just simply showing that using this virus, we're able to deliver a green protein. It's called green fluorescent protein. And sure enough, we can see green in the cells, the photoreceptors that matter. So it means, yes, this vector, this virus is delivering what we need to. And on the right-hand side, what we've done is we've used the gene in question, BBS1, and asked the question, and this is human BBS1 that we're delivering to the mouse, And we're asking the question, can this actually be expressed? In other words, is it being seen in the cells that matter? And yes, it is. So that's a good start. Now the question is, can we actually deliver this to the mice, the actual gene therapy itself, and make a difference in terms of finding benefit? And what we're able to see in a nutshell is that we can prevent or halt the retinal degeneration from getting any worse and preserve the cellular structure in the back of the eye as well. And so we perform OCT on these mice, and that's both the wild type mice, the normal mice, and the mice that have BBS. And we look at the back of the eye, and we can see with the medium dose on the left-hand side we're showing here, the thickness of the retinal layer, or what's called the outer nuclear layer. And certainly at 6 months, there is none left in the untreated mouse. There are no photoreceptors to be seen. With the medium dose, we see good preservation, but with the high dose, we see even better preservation here. And all I'm doing on the right-hand side is showing you graphs of the readouts from these. There's a health warning. There are several graphs, but they're really important. But importantly, what we're showing here is that this is what we call a durable treatment. In other words, when we get out to 6 months, we're still seeing this benefit of the thickness, maintained thickness and structure of the cells, of the retina rather. But if we look very closely at the end of the study, at a single mouse, for example, where we've looked at both of the eyes. So the right eye here is shown in B, and it's been treated with the medium dose of the therapy, whereas the left eye has not been treated. And you can see at the bottom here, what we're looking at is what's called this ONL, the number of cells. There's only about 2 cells thick, and there are no photoreceptors in that untreated eye, as we would expect. However, on the right-hand side, you're seeing in the area that this gene therapy has been injected, we're seeing complete preservation of the photoreceptor layer. So this is good evidence that this is working, but that is only as far as the cellular structure is concerned. What about the function? Can these mice actually receive light and send it to the back of the brain? And so we use ERG for this. I won't go into the details because it's quite complicated, but we essentially try to measure the function of the rods and the cones using different measures. And the graphs, those who like looking at graphs, simply the lines at the bottom here with the boxes is the untreated eye, whereas the other round circles are the treated eye, and you can clearly see a significant difference in the function at 6 months. And so not only do we see the preservation of the structure, but we're seeing preservation of retinal function as well. And then finally, we ask ourselves, after this 6-month initial trial period, are we still seeing that particular gene being expressed in the back of the eye? And sure enough, yes. And it's also dose dependent. So the graphs here, which are obviously quite complicated, but the ones that I've highlighted in red are the mutant mice that have been treated with the low dose, the medium dose, and the high dose. And importantly, they have significant expression. So in other words, there is still protein being expressed in the eyes of these mice after 6 months. So we expect this to have what we call long levels of durability. So some conclusions. So we know that gene therapy is highly effective. It treats the root cause, unlike all of the other therapies we've been talking about. Gene therapy still has a long way to go. It's been around for a long time. It's nothing new, but I can say that there are over 500 gene therapy trials currently underway. And again, it's something that I think we're going to see a lot more of in the next few years. There are probably around 25 or so BBS gene mutations, the most common one of which is BBS1, at least in the West. There are other countries such as the Middle East and Eastern Asia where maybe other genes are more prevalent. we believe that with the preclinical studies that I've demonstrated today, that not only is this gene therapy safe, but it's also effective. It's effective at preventing visual loss if given early enough. And then what are the next steps? Well, clinical trials. But the question for us is when, where, and how. And we don't have those answers just yet. So I want to thank the team who carried out all of this work, and particularly Victor, who not only designed the studies and oversaw and also interpreted all of the data from this. And I also want to thank Alsa Ventures, who have believed in us to be able to carry on the dream. And thank you very much.
Thank you so much, Phil. Some really exciting updates there, and no doubt I can see some questions coming in that we can put to you in the Q&A after the next speaker.